So our next and final speaker is Dr. Tracy MacGill, who is Director of the Medical Countermeasure Regulatory Science for FDA's Office of Counterterrorism and Emerging Threats and the Medical Countermeasures Initiative. And she's going to talk to us about strengthening regulatory science for pandemic preparedness.
So I'd like to thank the organizers for inviting me to speak today about FDA's Medical Countermeasures Initiative and strengthening regulatory science for pandemic preparedness. So this disclaimer basically says I'm speaking on my own, that the views are not expressed in this presentation are those of the presenter, and not necessarily do they represent those of the U.S. Food and Drug Administration, nor should they be interpreted as official agency policy. So the Medical Countermeasures Initiative was launched in 2010 after a year-long comprehensive cross-government review. And the goal of the program is to promote development and availability of safe and effective medical countermeasures. Medical countermeasures are defined as FDA-regulated products that may be used during a potential public health emergency. So the Medical Countermeasures Initiative has three kind of overarching components. One is legal, policy, and regulatory frameworks. One is regulatory science, and one is professional development. So the regulatory science, which I'm going to speak about today, the goal is really promoting the development and availability of safe and effective countermeasures, and by developing tools, standards, and approaches to assess medical countermeasure safety, efficacy, quality, and performance, and importantly, translating cutting-edge science and technology into innovative, safe, and effective countermeasures. So really, the goal of the program is to develop solutions to complex regulatory science challenges, which are things I know everyone here is familiar with. And a robust medical countermeasure regulatory science program creates the data necessary to support regulatory decision-making. And so the components of our program are intramural and extramural research. So intramural research is research conducted by FDA scientists. And the extramural research is research that's conducted by external partners, whether they are at other government institutions or universities, private research institutions. All of this research aligns with the U.S. government public health emergency medical countermeasure enterprise priorities. And we utilize vehicles that are managed through FDA's Office of the Chief Scientist. So the intramural program is facilitated through the FDA Challenge Grant Program. So it's an FDA-wide program that funds work, again, throughout FDA. And the Medical Countermeasure Intramural Program is just part of that overarching program. We also utilize FDA's Advancing Regulatory Science Broad Agency Announcement to fund our extramural projects. So this is an open solicitation to which people can apply, and I'll talk a little bit more about that later. And obviously, scientific collaborations are key to the success of the program. So since the beginning, our program, the initiative started in 2010, but the regulatory science program launched in 2011. And since that time, we have funded over 300 challenge grants through our intramural portfolio, and we have broad center participation. So all of the centers within FDA whose research is relevant to medical countermeasures have received awards. The extramural portfolio has supported 7 interagency agreements. Those are agreements across government to either work together or support research at another government institution, and 2 cooperative agreements. So the cooperative agreements are used to support our professional development programs. We have 2 trainings. One is for data quality and integrity in non-clinical studies for high consequence pathogens, and we also have one for data quality and integrity in clinical studies of high consequence pathogens. And those are completely free to attend. Those are held annually, both in person and with a virtual option. And so if you're interested in attending, please feel free to reach out. We are fortunate to have collaborations across the intra and extramural portfolios. And I think importantly, the portfolio, as you'll see, has been capabilities focused, and that's been a real advantage. And most of the portfolio in recent years, no surprise, has been focused on public health emergencies such as Ebola, Zika, and of course, SARS-CoV-2. So for this audience, I thought you'd be interested in learning a little bit more about the broad agency announcement. So this covers research priorities across the agency, medical countermeasures just being one of the many topic areas. And this is aligned to FDA's overarching scientific priority areas. It's a two-stage application process, and it results in a contract award. So this is a screenshot of the current announcement, but it is updated annually. And in the case of our program, we always encourage folks who are interested in applying to reach out to us and have a discussion and to ask about the mechanism itself before submitting an application. Obviously, once an application is submitted, it needs to go behind the wall through the official process that is managed by our acquisitions office. So again, one of the goals of the program is to develop regulatory science tools. And this slide is an example of some of the tools that have been developed through the support of the program. So we have the FDA Argos database for regulatory grade microbial sequences. And that has included sequences for Ebola, Zika, and SARS-CoV-2. And this was featured in an FDA news release. We also have supported the development of reference materials for diagnostic developers for Zika and SARS-CoV-2. We are supporting microphysiological systems models of acute radiation syndrome, Zika, and SARS-CoV-2, and a range of organ systems. We have supported a cross-species immune system reference database for Ebola, Zika, and SARS-CoV-2. And we've also worked with the CDC to develop an antimicrobial resistance isolate database. So we've really, again, our portfolio is capabilities based, and this has really enabled us to leverage our projects for public health response. And so in the next few slides, I'm going to walk you through some of those projects. So we initially had in 2015 and 2017, we had two projects with UKHSA and University of Liverpool focused on Ebola. And then early in 2020, we were able to leverage that project for SARS-CoV-2. Similarly, and I have to really give credit to Miles and Julian and their teams. This enabled us to do some of the very first of their kind studies during the pandemic. And if we had not had that project, we would not have had that opportunity. We also had a project with Stanford University, Dr. Gary Nolan, and looking at sequela and immunopathology in Ebola and Zika. And then we were able to expand that to include another capability, the MIBI. And this was co-sponsored by NIAID. And again, because we had that project and because of the capabilities of that project, very early in the pandemic, we were able to leverage that to answer some important questions about COVID. And then we had a follow-on award building on that work, again, with Stanford University. And then in 2021, we modified that project to identify biomarkers and immune correlates in clinically diverse populations. And that was co-sponsored by FDA's Office of Minority Health and Health Equity. So then, similarly to the Stanford project, we had a follow-on project with the University of Liverpool and UKHSA. And again, that was looking at characterization of viral evolution, host immunity, biomarkers of disease. And that was also co-sponsored by NIAID. We modified the project in 2021 to look at variants of concern. And then in 2022, we expanded that work. So we also had a project focused on Ebola awarded in 2018 with the Broad Institute. And they were doing spatiotemporal mapping of gene expression. And again, in 2021, similar to the other projects, because of the capabilities, we were able to do a mod to look at transcriptomics and single-cell RNA sequencing to better understand viral infection and immune responses. We have organ-on-chip work with the Beast Institute looking at acute radiation syndrome. So this was a 2015 project that was a follow-on of a 2012 project, which was co-sponsored by FDA's Office of Women's Health to develop female chips. And we were able to, in 2021, mod that contract to develop a human and non-human primate organ chip models of COVID-19 infection. And that was in collaboration with UKHSA, also co-sponsored by NIAID. We have a project, again, Ebola and Marburg focused with University of California, Los Angeles, Dr. Annie Ramoy. That was awarded in 2019 to establish a biobank of survivor samples. And then in 2021, we were able to expand that work to include development of a Marburg survivor biobank to inform vaccine development. And that is co-sponsored by the Defense Threat Reduction Agency in DoD. More recently, we awarded a project to Emblema and George Washington University for expanding next generation sequencing tools for pandemic preparedness and response. And really, they're developing quality matrix tools to expand FDA's Argos database, which is something we've been funding through the intramural program. And we have modified that project in recent years to just expand some of that work that we're doing. We also awarded a project to the Commonwealth Scientific and Industrial Research Organization, CSIRO, in Australia to strengthen coronavirus models with systems biology and machine learning. And so they're using genomic and multi-omics analyses and machine learning to understand responses in a range of non-clinical and clinical COVID-19 models. So some of the applications, and it's not limited to this, but these are some examples of things that have resulted from this work. Respirator decontamination for emergency reuse. In 2016, we funded 2 projects to look at the potential decontamination of respirators in event of a public health emergency. And so the first project was with Applied Research Associates, and they developed a consensus standard for UV surface decontamination in 2018. The second project was with Battelle, and they were using hydrogen peroxide vapor decontamination. And that ultimately led to the development of the critical care decontamination system, which received an EUA in March of 2020 for decontamination of masks. In 2016, we awarded a project to University of California, Davis to understand Zika distribution and duration for in tissues to inform donor screening. And that ultimately supported an FDA guidance in 2017. In 2018, again, one of the projects that I just talked about, assessing Ebola host response and medical countermeasure efficacy, again, because we had that project and we had those capabilities of the project, we were able to leverage it to do very early studies in the pandemic that ultimately helped to establish very important ferret and NHP models. And this was in collaboration with not only across government, with some public-private sector partners. Then in 2021, this was a result of our intramural work at FDA. That work informed an FDA guidance. So clearly, partnerships are very important to the success of the portfolio. And certainly the partnerships that we have with our other government agencies to co-fund, that I feel that means we're doing the right things in terms of being good stewards of our federal dollars and not being duplicative. And those sort of came about because we do work very closely through that FEMC mechanism, that we have the ability to talk across government in a variety of spaces, including regulatory science. And so in those cases, these were projects that came through the FDA BAA, and we said, well, we know someone who might be interested in this. They're working on similar questions. And then we discuss, is this something that you would be interested in helping to co-fund for both of our programs benefit? And one thing that I really would like to add about the BAA that's really, I think, very helpful is that sometimes when you see these solicitations, it's a statement of work that an agency needs. And they're asking the community to propose an approach to get to that end goal. The FDA BAA is not like that. It actually has a list of the topic areas across FDA that FDA is interested in, our needs, and we're asking the community to propose a project that they can do that would fill an identified regulatory science gap in that area. So as an applicant, you're not directly competing against another applicant trying to do the same type of work. You're helping us to identify gaps and proposing work that you could do to help us fill those gaps. And because of that, we've gotten amazing work that certainly I could have never thought of. And so I think that that's a real benefit to the program. So another type of partnership that we have is that recently we worked with NASA, HHS, and NIH to put out a joint solicitation looking for extended longevity of 3D tissues and MPS systems for modeling of acute and chronic exposures to stressors. So this was similar to the, I guess, more traditional solicitation. We got together and said, we have a common need. Let's put out a call for proposals to address this identified need. And really, it's extending that viability of those systems to 6 months that's going to broaden the potential applications of them. And so through that project, we funded 8 proposals that are listed here for a range of models. So clearly, scientific engagement is very important to the program, and we welcome outreach if you'd like to participate. And so in conclusion, investments in innovative tools and approaches may address important regulatory science gaps. Partnerships are important for maximizing investments, establishing networks that support medical countermeasure development. And these approaches can facilitate timely and efficient research response to emerging infectious disease threats, as has happened during the pandemic. So I have to acknowledge my team shown here. All the things that I've presented would not happen without them. We have a relatively small team, and they're the best. I'm very lucky. And for more information, my contact information is here, and the QR code should take you to our little flyer about our program if you'd like more information. Thank you.
Thank you, Tracy. Has anyone got any questions?
I'd like to ask a question if I can.
Go for
I
it.
can abuse chair privilege, and then Miles will get a turn. I was interested in the amount of modifications to the contracts where you I take it you were repurposing money rather than giving them extra money.
No, I mean, we have the ability to do a mod either way, right? Depending on the need, depending on the resources. So we can do a modification to within the overarching scope, right? If it's something totally different, we have what's in that statement of work, and you have to stay right within that. We would have the ability to say, look, we'd like you to do this additional work, kind of with what you have, efficiencies. However, for the most part, when we do these mods where, for example, we're utilizing a project focused on Ebola to now do SARS-CoV-2 work, that did come with funding to do that SARS-CoV-2 work as not to compromise the Ebola work that was initially awarded.
Okay. Well, I guess the actual specific question was, does that come from the scientists, or does it come from you, or does it come from both, depending on the circumstance?
You mean in terms of the direction, like the work?
Well, do you say to them, do you say to Julian and Miles, I want you to stop focusing on Ebola and focus on SARS-CoV-2 because it's more immediately important? Or do they go to you saying, we would like to do this, we think it's still within the overall remit of the grant, do we have your permission?
I mean, I think we are really lucky to have great working relationships across the portfolio, and we have regular communication. And so I would say it's a little bit of both, that, if we see something from our side that we feel, should be considered or needs to be done, we will have that conversation with our partners. If they see something from their side that, a gap or work that needs to be done, they would come to us. And then we have a conversation about, okay, is this something we mutually need that we can find a way to support? And is that something that is possible in the context of the existing projects you have? Or is that something that would require a new application through the BAA? And again, as you saw with some of that early pandemic work, we were somewhat limited, right? Because it had to be done quickly and those projects were focused on Ebola. And so we did what we could in the interest of time and available resources. And then, based on that information, there was more work to be done, at which point they came in with a new application that resulted in those standalone COVID-focused projects.
Actually, Paul, you covered the question I was going to ask, which was great. The key point is that when you're writing normal grant applications, it's more like just a basic science, and it's very much translational for the FDA needs in helping other people get product through the regulatory pathway. I think, Julian, you had a question?
Thank you. So, Tracy, lovely summary of your activities in your team. And it was kind of around what Paul was saying. And as a funder and as a regulator, how important do you think it is to be responsive to something new? So again, in the UK, I was saying to Nikki, and our funders in the UK responded pretty quickly to the outbreak and having a call, but you guys were almost instantaneous. So do you see as funders that pre-positioning and that confidence in teams as being important?
No, absolutely. I think we try to make the best investments that we can as any funding agency. And really, we were just very, very fortunate that the projects that we had invested in were capabilities that could be leveraged, and the PIs and their teams had that expertise. And so really what we did was we just recognized that opportunity. And again, as noted, that's why it's so important that we have the open, constant communication with our performers, because that really, I think, helps us understand, evolving needs in a public health emergency and thinking about, okay, how can we be part of the solution? And that was something that I tried to stress to my team. I said, this is obviously, it's a terrible situation the world has found itself in. However, and everybody's locked in their houses. However, we have the opportunity to be part of the solution versus being locked in our houses and hoping that other people will solve it.
And just to comment on that, in the UK system, the institute system was something that was in place and could be very rapidly repurposed towards the outbreak problem. So in our own sphere of virology, the MRC Institute Centre for Virus Research and the BBSRC Pirbright and Rosamond Institutes just swapped straight away into doing SARS-CoV-2 work. But I must admit, in my own little domain, we did it, and then we remembered to ask for permission. Nikki, I can't see you in the audience, but yeah. We did remember.
Yeah, thank you, Tracy. I just wondered which US funding stream funded the PREDICT program, because I don't think it was FDA, was it?
Yeah, I'm not sure, but I was likely, through the public health emergency medical countermeasures. Yeah, I think you're right.
As a regulator and a funder, what do you think is the biggest gap that needs to be filled the next time we're in an emergency?
That's a good question. It's a hard question. And so I think I'm not in the regulatory side any longer, so I can't really answer for them. And I really commend my colleagues who were in that side of FDA. They worked tirelessly And really, they reviewed things and obviously the EUA process. I mean, they really are to be commended for their hard work during the pandemic. I think from a regulatory science perspective, I think, again, it goes back to for me, the advantages of having capabilities. And that does not mean it's not important to focus on very specific questions in a traditional way. We need to get the data to fill the gaps, whatever the best way is. However, I think in our program, because we were fortunate that we were investing in capabilities to answer those programs, then when we really needed it, and we really needed to be able to have a research response, those capabilities, I think, well positioned us to do that. I think also, again, we're not a traditional funding agency, so we don't understandably have a traditional funding agency, budget. And so again, I think capabilities also helps us to really maximize what we're getting for our research investments.
Thank you for having so much organ-on-chip content in the slide deck. And I just wondered whether you wanted to comment on the FDA Modernization Act that was recently approved in US Congress.
Sure. So yeah, and actually I'm glad you mentioned that, because FDA-wide, there are, several groups that are working on alternatives. We have an Animal Welfare Council that, they play an important role. We have the FDA Alternative Methods Work Group. These are cross-cutting groups. And for example, that group has the ability for folks to come in a non-regulatory environment to essentially give a seminar presentation of an alternative method that they are working on. And there's, through their website, you can come in and say, this is our organization, this is the model, we'd like to talk to you about it. And so you have folks from across the agency, regulatory centers, but having a strictly scientific conversation. And so I think, that's a really nice mechanism. We have other overarching alternatives, activities, at the FDA. So I think it's an acknowledgement, right, that alternatives are an area of great interest. And so the FDA has really responded in that way. As you note, the FDA Modernization Act now allows for the potential use of alternatives.
Okay, maybe one more question.
Callum Semple in Liverpool. This is a blatant advert. The success of the ISRIC method of operation in COVID led to a little known group in the UK Health Security Agency approaching us to say, look, this worked really well for biological infection. Is there any reason why we couldn't change it for all threats, by which they meant CBRNE? And it turned out that the ethics committee were happy for us to change the word infection to exposure. And in doing so, exposure encompasses chemical, biological, radiological, nuclear, and interestingly, E, which is energy. And energy's moved from radiological and nuclear to potentially including unexplicable energy exposure, such as infrasound or focused microwave energy. So the ISRIC protocol is now available and could be used by you if you wanted to rapidly recruit a group of returning diplomats. You could then use that framework to take their samples into a research environment. And I think that might be a useful tool for your medical countermeasures. It's certainly being used but certainly available for use. I think I get a red dot on my chest if I said it was being used for this kind of environment. So at least something good has come out of COVID. We've learned how to use these tools better. Thank you.
Absolutely. Thank you for that. That's a very good suggestion.
Okay, thank you for your questions. I think with that, we'll just thank Tracy again.
Actually, we should thank all the speakers for excellent talks and for keeping to time.